I figure let's start now, and then it's only the intro bit, so people can then just join in. So I'm gonna like formal face now. So yeah, firstly, thank you so much everyone that's logged in and joined in. I'm really excited for this. I'm really sure that you're going to really enjoy it as well. So this idea came to me after joining OHP Society and being graciously offered the post of running the sort of research division, despite my I have a degree, but that's about it. I've got no papers to my name, unfortunately. But it was really, I just felt like it was really needed in the med school, some kind of informal teaching about how research works, the different aspects of it. You know, we kind of walk in and we're expected to know how you read a research paper, how you make a scientific poster, all these different things. And I feel like it's something that's not properly taught. So I wanted to give students an opportunity to learn that from the people doing it and have done it. Which obviously led me to talk to Ben. For those of you that don't know, Ben, Dr Towler, he was my studentship, my summer studentship and my dissertation supervisor in the lab. So he coached me through my third year basically, and gave me a lot of support, which is why obviously he was my first thought for this, because he explains things really well. He's not patronizing. And also it can be a bit intimidating, the professors, to like feel you can ask some of the sillier questions as well. So that's kind of where I was going with. And sorry, I'm just trying to find my notes as well, because I wanted to basically big up Dr. Towler, because hang on just a sec for me. I know I should have this all sorted out, but hey, there you go. Such is the student life. There we go. So just to carry on from what I was saying, so Dr. Towler, he had his undergrad in biochemistry and then did his PhD in molecular biology and genetics, graduating in 2017, which I think is around the time when we met. Is that right?
Yeah, I think I was just coming to the end of it.
Yeah, that sounds about right. So he's been a postdoctoral researcher in Sarah Newbury's lab in the medical research building, which for those of you who don't know is attached to BSMS. It's like the building just behind the med school building on Sussex campus. So the work that they do in the Newbury lab is all about RNA stability and degradation. So working on gene expression in proliferation and tissue growth. Dr. Towler's also organised RNA biology grouping at Sussex. He's authored 11 publications since 2015 with 185 citations, as well as winning national and international awards as well. So he's basically a local hero. So we're really lucky to have him here to talk to us this evening. I'm going to stop rambling now because that's boring, and I'm going to hand over to Dr. Towler. So take it away, Ben.
So if I share my screen, if you can let me know that it's working.
Sure thing.
So can we see the presentation?
Yes, we can.
Yeah, brilliant. Cool. So yeah, thank you, Evie, for the introduction. So as Evie said, I actually think this is a really good idea to sort of get you to talk to us, really, because we are here, we're in this, as Evie said, we're in the building that looks like this, which is attached to BSMS and JMS. You will only really see this if either one of your sort of academic skills tutorials have any of the lecturers that are situated in the building. Other than that, you probably don't really know we exist. But yeah, as Evie said, my scientific background, I did a BSc at the University of Sussex in biochemistry. And it was during that time that I really sort of fell in love with science. During a project with Professor Sarah Newbury, who I'm still with. So for the last 8 years, I've been working in her group. So I thought the way I'm going to structure this talk in general is I'll talk a little bit about why I chose scientific research, the work we do, so give you an overview of largely speaking the data that we're in the process of publishing. So I'll give you an overview of that, and then we'll go into sort of how I look at reading a scientific paper and how I try to critique it and give you some sort of tips from my experience on how to get the most out of a paper and then leave it open to any questions that you want to ask me, whether it be about how to read a paper, about science itself. And so, yeah, so feel free to ask as many questions as you like at the end. Not a problem at all. So this is our lab here. So this is my bench. We obviously have the microscopes and all the kit and everything. Evie, you probably are familiar and recognize it from your time here. Hasn't really changed that much since. So why did I go into scientific research? So while I was at school, so secondary school and college, I had some really fantastic science teachers that sort of built on my fascination of how things work. So science is obviously the understanding of how anything works within your cell. I found a presentation online actually, which I thought this sums it up really nicely. And science is a way of thinking. It changes how you look at the world. And what it does is it gives you the skills to question what people tell you. And again, I think this is a really important thing when we get onto to how to read a scientific paper later. Yeah, so as I just mentioned, my work experience that I gained during my undergraduate degree really is what made me realize that science is what I love. And it allowed me to follow my curiosity. Ultimately, we're all similar, whether you're a medic or a researcher, we're filled with this curiosity of how things work in the cell and why, when they go wrong, can they result in such fundamental things. And ultimately, I think the big two things with me with science is that you never know what you're going to find, right? We ask these questions and we never know whether these questions are correct, and we just try and answer them as logically as we can. And ultimately, whatever we see when I look down that microscope or when I look at any gene expression profiling, I'm the first person, hopefully, to have ever seen that. And I think that's pretty cool. And that really is what keeps me going. So what am I interested in? So as many of you will be aware, obviously within your cell, here, so we have our nucleus, and then within the nucleus, we have our DNA. Okay? And the central dogma of biology explains the journey of gene expression effectively from DNA locked within this nucleus, which is then transcribed into RNA. So this is almost a mobile messenger. So this is how we get from our blueprint into our functional product. So this then moves out of the nucleus into the cytoplasm, where it is made into protein. And these are the functional building blocks of our cells, amongst a lot of other things. But what is important during this process, you would imagine that to get from here to here, the cell has to be very, very accurate in what it's producing. So we can't have steps going wrong during transcription, during translation, which is the process of making RNA into protein, and during protein function. So there's all these different levels of regulation that go on during this journey to make sure that your cell functions correctly and we don't get disease. So what I'm interested in is this step here. And so this is the controlled degradation of mRNA or any RNA species. And why do mutations or mistakes within this degradation pathway result in disease? And so what I like to think about is the amount of RNA within the cell is a constant balance between the amount that's synthesized by that process of transcription, so the reading of DNA into the mobile messenger, and then how that messenger is removed. Because you might imagine if there's too much of this around, if it encodes a protein such as an oncogene, so you might have heard of MYC, for example, which is a very classical oncogene. If it makes too much of that protein, then you can get cancer, for example. And so it's really important that the amount of mRNA is tightly controlled. And so this is what I'm interested in. And so to give you a very brief overview of how this happens. So normally, so we're taking, again, this mRNA, so our messenger, which is made into the functional protein, is protected by structures at the 3' end. So RNA is polarized. So we call the left-hand side the 5' end, and the right-hand side the 3' end. And we have these protective structures called the poly-A tail and the 5' cap. And these stop the enzymes which are involved in chewing up the RNA. So these are called ribonucleases, from accessing it. However, when the cell wants to remove this RNA from the cell to make sure it doesn't make too much protein, we get a process called deadenylation. So this removes this poly-A tail, and then this leaves a vulnerable end, which either complexes such as the exosome and a catalytic subunit DIS3 or DIS3-like 2 can access and then chew up this RNA and get rid of it. Alternatively, we can get removal of that protective cap structure by a complex called the decapping complex, and this then leads to degradation in the opposite direction by a protein called XRM1, which is known as Pac-Man in Drosophila, or fruit flies. Because naming in fruit flies is far more fun than in humans. But the important thing about this is each one of these enzymes are involved in different diseases. So for example, if you look at DIS3, so this is this catalytic subunit. So this is the thing that actually chews up the RNA. This is mutated in multiple cases of multiple myeloma and acute like myeloid leukemia, so hematological cancers. If we look at dys3-like-2, which is the gene that I work on, which I'm going to talk to you about a lot more, is that this is involved in overgrowth syndromes known as Perlman syndrome and a kidney tumor known as Wilms tumor. And it's not just these proteins that are involved, XRM1 is also involved. And for example, quite topical at the moment, a lot of RNA viruses have been shown, they haven't looked at the COVID virus yet, but a lot of RNA viruses have been shown to have a really neat mechanism. They inhibit XRM1 to allow their viral genome to survive and encode a number of, and basically enable them to survive and propagate. Reduced levels of XRM1 are also observed in osteosarcoma, so this is another paper that we currently have in process. So as I mentioned, I'm interested in dys3-like-2. So dys3-like-2 chews up RNA, and it's called an exoribonuclease. This basically means it chops up ribonucleotides from the end, right? So this is the naming, and it's very, very, very highly conserved. So from bacteria all the way through to humans, we can see here that this is the domain structure of the protein. And so, for example, the bit that does the chopping up is called the RNB domain, and this is this orange domain. So you can see this is to scale. So the size of this region is very, very, very well conserved, which means that it's a very important protein. Evolution has selected it for whatever function that it has within the cell. It acts specifically within the cytoplasm. And as I mentioned, it's very, very highly conserved, and to say that family member as far back as bacteria, and it does exactly the same job in bacteria as it does in our cells. And as I mentioned, loss of function mutations result in overgrowth syndromes known as Perlman syndrome, which is here, which I'm going to talk to you about a bit more in a second. And also deletions of dys3-like-2 have been observed in up to 30% of sporadic Wilms tumor samples. So again, this is this kidney tumor. So this is a mouse kidney here. So on the left, you have a normal kidney. You see Wilms tumor is massive overgrowth. So Perlman syndrome is a very rare disorder. It's generally autosomal recessive. So this means you need, you have 240 copies of dys3-like-2 in any chromosome that isn't your sex chromosomes. And it's been shown here that you have deletions. So this is a paper by Astuti et al. That came out in 2020. It's a really nice paper, which showed that in here you have bits missing of dys3-like-2. So that's all they're looking at here. So you can see the absence of this band shows that there's bits missing. So the prognosis is very, very poor for this disease. There's only 5 reported cases where the child lives beyond 1 year old. So it's a very, very severe disorder. And its clinical features are characterized by sort of large birth size, known as macrosomia, macrocephaly, which is enlargement of the head, enlarged abdominal organs, which is known as visceromegaly. So you can see here how swollen this abdomen is. It really shows how big these organs are growing inside. We also get hypertrophy of the islets of Langerhans, skeletal abnormalities. And these patients, if they live long enough, they often show a predisposition to Wilms tumor, which is that kidney tumor that I mentioned. And so it's been shown that all Perlman syndrome patients that have ever been discovered have mutations in dys3-like-2. So it's really clear that dys3-like-2 is important for tissue growth and correct development. So as I mentioned, they also develop Wilms tumor. And so Wilms tumor is the most common childhood kidney tumor in the UK, with between 80 and 85 cases per year in this country. It generally affects young children aged 3 to 4. The incidence of it drops off after the age of 5. But it's interesting that only 1% cases are genetic. So this would be a hereditary sort of mutation, whereas up to 30% of sporadic cases have deletions in dys3-like-2. And these are normally very, very big deletions. So some of them wipe out the entire gene. So it's thought to be caused by clusters of primitive kidney cells that are normally involved in kidney development during embryogenesis, that normally disappear at birth. However, they stick around, and then what you get are these big growths on the kidney, as you can see here. So symptoms for Wilms tumor include swollen abdomen or abdominal masses because of this swelling on these growths on the kidney, blood in the urine, and then standard fever, weight loss, lack of appetite, upset stomach. So quite general symptoms. And treatment currently involves surgery. So it depends. Sometimes the very severe cases, you can get tumours on both kidneys, but while there's on one, then surgery is an option, and then classical chemotherapy and often radiotherapy is used to try and shrink it enough to then undergo surgery. So both of these overgrowth syndromes are caused, at least in either completely or at least in part in Wilms tumor case, by mutations in this single enzyme, which is involved in controlling the amount of RNA within the cell. And so this brings me on to sort of the main work that I do in the lab, which is understanding the role of dysrelate-2 in these diseases which are caused by excess proliferation. So the main questions that I have within the lab are, what specific RNAs is dysrelate-2 responsible for creating? So I showed you earlier that these different enzymes which chew up the RNA are implicated in different diseases. So this tells us that they have very specific roles within the cell. Otherwise, you'd expect the same thing going wrong would cause the same disease. So we know that there are specific groups of RNAs which dysrelate-2 is told to remove. So what we're interested in is what the identity of those RNAs and how dysrelate-2 is specifically told to chew up that RNA at that certain time within a cell's life. And what processes these RNAs are involved in. So for example, what proteins do they encode? And then what's the result of increased expression of those proteins, for example. And then finally, move on to a more clinical setting, which is what of those targets are also misexpressed in these human conditions that result in this disease, and how does this drive disease? And this leads us to our hypothesis, which I'll talk to you a lot about when we're going through the papers, which is the most important thing of any paper or any scientific procedure is to be hypothesis-led and to make that hypothesis incredibly clear to your reader or your audience. And our hypothesis is that dysrelate-2 specifically regulates a subset of RNA transcripts, which in its absence become misexpressed and drive Pellman syndrome and Wilms tumor. And so this is the question that we've tried to answer in the paper that I'm going to sort of go through with you now. So we have used two different things. We've used Drosophila, which is sort of the humble fruit fly, which again, our host Evie helped me with when she was in our lab. And so these are really, really good model organisms to study proliferative diseases. We use these and human cells. So Drosophila itself has been used as a model organism for more than a century. They're very cheap in terms of model organisms go, and they're very easy to keep. So they're also very easy to genetically manipulate. So the interesting thing is the Drosophila genome is much, much smaller than the human genome. It only has, because of introns and things like that, there's much shorter introns, and it's only got 4 chromosomes, but actually the number of genes that are encoded within the fly is very, very similar. And what's most important is the pathways that control how our cells behave and how our body develops are incredibly similar and very, very well conserved in humans. So for example, if we look here, the way our body is organized, the way our digestive system is organized, our nervous system, the fundamental building blocks of these critical symptoms are incredibly similar between the fly and the human. And so this means that what we find in the fly, we're able to relate to humans and understand much better. And this is shown again by up to 75% of all disease-causing genes have been shown to have a functional homologue. So this means that they have a protein that does exactly the same job in the fly. And it allows us to study the role of dysrelate-2 in its natural context. So we also use human kidney cells. So this is a picture of kidney cells. They grow in sort of clumps, but these are just individual cells growing in a dish, right? And so they have their place. However, it's much better to also look in a natural system, which haven't been told to grow indefinitely, which is what these cells do. And Drosophila have played very important roles in the development of a number of anti-cancer drugs. So methotrexate is known as a workhorse of cancer treatment quite often, and a number of other treatments as well. So the way methotrexate functions was discovered in Drosophila, and also vendetinib. So the Drosophila is a really, really good model organism to try and study the role of this enzyme in its natural setting. So what we did, come on, what? Oh, it's completely, sorry, everything's just gone off my slides. I'm just going to come out the presentation and see if it's, sorry about this. It was working absolutely fine a minute ago. The joys of technology.
We did do a test.
We did. what I'll do is I'll close it and reopen it.
That's fine, don't worry.
No,
just while we have this pause, if anyone has any questions, you can put them in the Q&A box, and what we'll do is right at the end, we'll go through them. So if you can put yourself as an anonymous, if you don't want people to know your name, or if you want to say it live, unanonymize yourself so that I can basically bring you up so you can ask your question in person. Okay, looks like we're back up. So
yeah, we're back working. Okay, so that's good. Sorry about that. I have no idea what happened. The computer did make a funky noise, so I don't know whether it tried to do something and then sort of. Anyway, so where were we? So as I say, what we want to do is use Drosophila to study the role of dysrelate 2 within proliferation, and then hopefully that'll help us understand the human disorders. So what we do is we use an approach called reverse genetics. And so this is where we remove the gene of interest. We have a look at what happens and then what phenotypes we see. So whatever happens to the organism, we associate with the enzyme, the gene that we've removed. So what we did is we use CRISPR-Cas9, which a number of you may have heard of, this big sort of, when they decide who owns the rights to it, they will win the Nobel Prize, I'm sure. It's completely changed the way we are able to do this sort of work. So what we did is so this is our gene with the 5' end that I mentioned on the left-hand side and the 3' on the right. And what we did is we removed this region here. So this is 8 base pairs that we removed, and this was enough to change the translation of the protein, meaning that we don't get any. So here we're able to look at the amount of protein within the fly, and we can see this band here is dysrelate 2 protein. But in our mutant, we don't have any protein at all. So we're able to remove dysrelate 2 throughout the fly. Strikingly, what we saw when we looked at the wing, so we used the wing as a model because the flies don't need their wings, for example. If we do something which is really critical to them, the fly can still develop normally. But what we see here is if we look at our control size, so this has normal dysrelate 2 sequence and normal dysrelate 2 protein, we can see that we get significant overgrowth. So this is the wing area on the y-axis, and we can see that the red bars, which are the mutants, are significantly larger than the blue. Okay, and if we do an overlay here, we see that the blue wing, the control wing, is much smaller than the red wing. And what this is showing us is that actually, if you go back to these human conditions, what we see is overgrowth. Okay, and that's exactly what we're seeing in the fly. So this is telling us that our model is good to try and study the role of dysrelate 2 within this setting. So what we were able to do is go into the precursor tissues, so the life cycle of the fly. We have the fly lays the embryo, we have a larval stage where all of the tissues are basically growing, and then they're defined, they're called wing imaginal or imaginal discs, and the wing imaginal disc contains all the cells that form the wing. And what we're able to do, we're able to count the number of cells that are actively dividing. And so our question here is, are the cells bigger because the cells are dividing more, or are the cells bigger themselves? So each red dot here is a dividing cell, and you can see in the tissue on the right, you can see it's larger as well than the one on the left, the control, but you can see there's so many more red dots, and this is telling us that the cells are dividing far more. And one cool thing you can do, hopefully this will work, is you can actually label cells at different stages of the cell cycle. So you may well be familiar with the cell cycle. So we go through mitosis, a growth phase, DNA synthesis, where your DNA is replicated, and then your G2 is a growth phase. So what we're able to do here is label cells at different stages of the cell cycle. So you see some yellow cells, some green cells, some red cells, and we can have a look at how the cells are cycling. So this is another thing that we're able to do in vivo in these live tissues. So what we wanted to ask, as I mentioned, going back to our hypothesis, what RNAs do dysrelate 2 degrade? Okay, so we're able to look using a process called RNA sequencing. We're able to look at every single RNA molecule that is expressed within these tissues. Okay, so we remove the RNA from these tissues, and we're able to compare effectively gene expression profiles, so which genes are on and which genes are off, between this tissue and the control tissue. And what we actually identified was a number of genes. So we saw 344 genes were increased in expression and 157 were down. And we found a number of growth factors which were increased in our tissues, which makes sense given our phenotype. And we're able to use Drosophila genetics. I won't explain this too much. It's quite complicated genetics, but what we're able to do is we're able to remove each one of these growth factors within our mutant tissue and see if it rescues the phenotype. So you would expect, for example, if we remove dystrophy-like 2, whatever is causing the phenotype, if we also remove that, we should recover the phenotype. We should look at, it should look like what it does under control settings. And what was really striking is that if you look at the blue bar here, so just concentrate on this blue bar and the red bar. So the blue bar's control, again, the red bar's mutant, so you can see it's bigger, wing area on the left. However, when we remove IDGF2, so this is a specific growth factor, you can see that the size, while these tissues don't have dystrophy-like 2, they also, when we remove IDGF2, we completely lose the effect of the loss of dystrophy-like 2. So this is telling us that this protein is involved in driving the overgrowth phenotype. And so these are a family of chitinase-like proteins. So they're highly conserved as well. So humans also have them. So they're called chitinase-like 1 or chitinase 3-like 2 in humans, or also known as YKL40 or YKL39. And they function to promote cell growth through a number of survival signaling pathways, such as AKT and the ERK pathway, which are very classical signaling pathways. And these are implicated also in human disease. So for example, if you look at renal cancer, which again is interesting given that we have an identification of dystrophy-like 2 in Wilms tumor, the kidney tumor, or gliomas, you can see in blue, you have high levels of chitinase-like 2. This is chitinase-like 2, or high levels in pink, of chitinase-like 2, and low levels in blue. So you can see if you have high levels of these proteins, your prognosis is very poor. So this is survival along here in years. This is taken from the Human Protein Atlas. So these proteins, which are homologous to the factor that we know is driving the growth in flies, are increasing a number of cancers. They're involved in a number of inflammatory diseases, including asthma, and they're also potentially been associated with Alzheimer's. So these are quite interesting proteins in their own. So what we wanted to do, I mentioned earlier that we want to use the fly and then we want to take that into human cells to see if what we're seeing is conserved. So strikingly, we did that, and we went into those kidney cells that I showed you earlier, that picture of the cells in the dish. And what we do when we remove dystrophy-like 2 from these kidney cells, we see exactly the same thing. So what we have here is a count of the number of cells over time. And the red bar has no dystrophy-like 2, and the blue bar is normal. So this is a wild type. So this is the normal growth of these cells. However, when we remove dystrophy-like 2, we see increased growth in these cells. And then we went, because chitinase 3-like 1 and 3-like 2 are involved in these two pathways, we asked, are either of these pathways misregulated? So are they increased in their activity in these tissues? And is that why we're seeing the overgrowth? So what we had a look at is, so these proteins are all activated by phosphorylation. So this is a modification which activates the protein. And when we see that, we see much stronger bands here compared in the knockdown compared to the control, and this is quantified here. So we see increased activity of these pathways. So if we look at this pathway, what happens, this is a very simplistic view of the pathway, is that we see a growth factor will bind to the receptor. We then get activation of a protein called PI3 kinase, which then a downstream activates AKT, which is one of the things that we looked at, and this activates mTOR, which is a real sort of key protein in survival and growth. And if you've done any sort of signaling in the past, you would have heard about it. So what we can do is, in a similar way to how we took away IDGF2, we can inhibit using drugs. So we add drugs to the cell to inhibit PI3 kinase, which then would mean that this activation should get lost. Okay, and with the hypothesis again that if we lose the activity of this pathway, do we stop the overgrowth phenotypes that is caused when we lose dystrophy-like 2? So what we do when we treat that, so I'll talk you through this, is, so again, in the solid line, so in the red here, we have dystrophy-like 2 under normal conditions, and here we have a vehicle control. So I'm going to, again, when we go through how to sort of look at papers later, it's really important to have negative controls. So what we want to know is that this drug is dissolved in something called DMSO. Okay, so what we want to know is adding just the DMSO on its own, is that having the effect, or is it the drug itself that's having the effect? And so this is a negative control to make sure what we're seeing is real. So under normal conditions with this DMSO, we can see the increased growth versus the control in this solid blue line here. However, when we add the drug to the scrambled control, we don't see any difference. So the blue lines are very similar to each other. However, when we add that drug specifically to the knockdown cells, we see that this growth is lost. So you can see the blue and the red dotted lines are very, very similar now. And so this is telling us that this pathway is involved in driving the proliferation phenotype. And so one of the questions we're asking is, could this pathway be upregulated in the dystrophy-like-2 mutant Wilms tumor samples, for example? Is this pathway driving the disease phenotypes that we see? So at the moment, I have to point out, we don't know that this is chitinase like 1 or chitinase 3 like 2. So we're looking at that at the moment. It could be something else that's activating it, but it makes sense at the moment. And so that's another few years worth of work to try and work that out. So effectively, we've just been awarded a grant from the BBSRC, which is really exciting. So we've got another 3 years worth of work on this. And one of the things we want to do in this grant is to look at the RNA targets in human cells. So like we've looked in the fly, we want to look at in the human cells to see what RNAs are being misexpressed and ask the question, are chitinase-like proteins also responsible for human cell overgrowth? We've also got a collaboration with a couple of clinicians and the Childhood Cancer and Leukemia Group, who are going to give us samples that are Wilms tumor samples, so patient samples. We're going to look at the dystrophy-like-2 status within them. And in those that have dystrophy-like-2 mutations, we're going to see if what we find in our kidney cells in a dish are also misexpressed within these patient samples. So this is trying to put a clinical setting on what we're actually working on. And so this is where this is going. So hopefully this paper is currently available on bioRxiv, which I'm going to talk to you about in a sec. And hopefully will be published in the next sort of month or so. So that's been the last sort of 3-4 years worth of work, which Evie did help with as well, I must say. She did some of the work in the fly for me. So that's sort of what I do and what we're interested in within the lab. And we're also interested in the stress response and how RNA stability is regulating the stress response, which is another avenue of work we're doing at the moment, which is looking really, really cool and interesting. So I'm happy to talk to anyone about that if they have any questions. So that's kind of a background into what I do and sort of the work that we're currently doing and some real life science, as it were. But obviously, it's all well and good doing this real life science, but what's important is that we're able to communicate that with anyone who's interested in reading it. Okay. And so you could be the best scientist in the world. If you can't communicate your science, you don't get anywhere, because you're judged on your science and your science communication in the same way that you would be as a medic, you'd be judged on how you are with patients. It's exactly the same situation. So I'll go through sort of the fundamentals of what goes into a scientific paper and the types of papers that there are, and then sort of give you some checklists on how you can critique a paper and decide if you believe what the paper is saying, and then leave you with some sort of tips at the end of it to see if you can then go back to and see if you find them interesting. Hopefully you do, to at least some extent anyway. So obviously we see these papers. So this is one of ours, our previous paper that we published in 2016. And this is that paper that I told you about, the Astuti et al., which is about when they first identified dystrophy-like-2 mutations in Perlman syndrome and Wilms tumor. So just a pretty picture, really. So there are lots of different types of papers. These are definitely not all of the papers that exist. These are the papers that you may probably come across the most. So they're original science papers. So these are sort of the paper that I would write, for example. So these are a classical science paper that demonstrates an advance in our knowledge of a specific subject area. And so this is, you might say basic science often goes into an original science paper. So this might be what your professors give you to sort of say, read this will give you some background on the work, or read this and make it into a poster, which I know happens as well because I've marked them. So you will have to sort of do this if you haven't done it yet. You also get clinical case reports, which again, obviously being a basic scientist, I'm not as familiar with. So these are examples of symptoms diagnosis, for example, treatment and follow-up of specific patients. And these are often as a result of new symptoms or side effects or drug treatments. You also get clinical trial reports, which again, as I say, I'm not as familiar with. I'm not going to talk about that because I haven't got the familiarity with it. But it's basically a paper outlining the results of a clinical trial. And what I will point out, if you're interested in this, there are consort guidelines which are used to assess paper quality in the clinical trial. So every clinical trial that is ever written, any report has to go along with the consort guidelines. So this is a good way to make sure that what you're reading is good and has been produced properly. And you also get reviews. So there's different types of reviews, but these are a critical summary of data. And if you're new to an area of research, then these are a really great overview they give you an overview of the subject area. So you might have just come into a lab to do a placement, for example, and you don't have a clue what they do. So reviews are a really good place to get an overview of the sort of knowledge that is known within that field at the time. You also get systematic reviews, which are a more structured format, which are normally answering specific questions as well. So there's a number of other papers as well, but say these are sort of 4 main ones. So what does it take to get science published? So as I mentioned, there often years of work goes into every paper that you read. So for example, it was really disappointing when I had about 2 years worth of work goes into about 2 lines in a paper. So normally there's a lot of work that goes into these papers and a lot of people that contribute to the papers, and you'll see that by the authorship. So what happens is you decide that your story, like we have at the moment, we've decided that we've got a relatively good, complete story. So we can say, okay, well, we're going to draw a line here and then we're going to continue it. And maybe that's going to be the next paper. Okay, so that's often one of the hardest things to do, because you get so obsessed with your work and you just want to keep pushing it, but you have to know when is the time to at least get that out. And then let's carry on working on it so you can show people what you're doing and people can see what you're doing, because it's not just about you, it's more about your work will help someone else in your field understand maybe what they're looking at a lot better. So I learn a lot from all the other researchers in our field, and I get ideas from reading their papers, and that's where the creative thinking really comes from in science. So what you do, you pick your journal. So this will depend on your field, how good your story is. You'll often hear the word impact factor, which I absolutely despise, but that's still what people use. And that's the number of citations that the average paper will get within that journal. So you send it to the journal when it goes under editorial consideration. Okay, so what will happen is the editor will have a look at your paper. They'll decide, is it suitable for their journal? Is it the right field? Is it asking the questions that they're interested in publishing? And has it been done well enough just by a quick overview of the paper? And if they decide no, they'll send it back to you and they'll say, sorry, whilst this might be interesting, it's not really of interest to us, so please try somewhere else. Alternatively, it gets sent for peer review. And so this is the most important process of scientific publication. So this gets sent to experts. So this is you normally get at least 3 reviewers. So these are experts in your field. Or what the editor will do is look at the work you're doing and they'll pick the experts that they want to send this to. And their job is to make sure that you've done your work correctly, you've done it thoroughly, and you've ticked boxes, which I'm going to talk to you about in a bit. And so what happens is they then read it. They then send their comments back. They often ask you to do extra experiments to firm up your hypothesis, for example. Or if they're unsure on a certain area, they'll ask you to clarify it within the text or experimentally. And then what happens is these comments get sent back to you. You answer their comments. You often do further lab work to address these comments. And then you resubmit to the journal. And then the editor will look at this. They'll normally send it back to the reviewers. The reviewers will then say, okay, yes, they've answered all our questions. Or they'll say, actually, no, they haven't answered this question. Can you ask them to do that, please? And then this can go, you normally only get this happen once. But occasionally you get the reviewers coming back and you keep going backwards and forwards for a little bit. So this can happen a number of times until they're happy with the paper. And then finally, the editor will send you an email, which you've been waiting for about 6 months, and say, congratulations, we like your work, we're going to publish it for you. So you have this celebration relief sort of feeling. But it's great. It means that then the paper is out there and people can read your work. People can use their work to then further their own understanding of the world. And so this is how any paper that you read will go through this very rigorous process. So there's a lot of work and pain and effort and joy at the end of getting any paper that you read into the public domain. So I'm going to talk to you about these original science papers because this is what I have experience in. So the sections of an original science paper, you'll see they have an abstract, which is a summary of their findings and what they've done. And this is sort of to grab anyone's attention. So we'll often read a paper based on the title and the abstract. You'll see if it's relevant to what your interests are, or if you want to read it any further. So that's used to grab people's attention. You have an introduction where they introduce the findings, the field, knowledge within the field and what question they're asking. The methods, which is often very, very specific and technical, with how they've done the work, their results, and then the discussion puts it into the bigger picture. Okay, so you would have done this if you'd ever written a dissertation or anything. It'll have exactly the same format. And so the way I like to read a paper is I like to read it for the first time is I briefly read the paper to get an overview of the work. So what I want to do from my first read of a paper is to identify the hypothesis. So the biggest thing you need to understand when you read a paper is what is the big question that the group are asking? Because if you don't understand that, then nothing's going to be in the context at all. And then I always, if I'm new to an area, I'll concentrate on the introduction, especially if you don't know a lot of the field, a lot of any research within the field. So if you've been given, for example, a paper by your lecturer to say, read this, you might not know anything about that field. And the introduction is really important to understand where their big question sits. Okay, so this will help you put this into context. And then I like to have a basic understanding of their approach. So identify their hypothesis and then how have they tried to address this? Okay, and you can use the abstract for this. As I said, this is an abstract from our most recent paper that I said is we're currently in the process of publishing. So we'll give a bit of background, we'll say how we did it using RNA sequencing, and then we'll summarize our results, our key results. And so you can use this to sort of understand kind of where the author's going. But then these are what I find is most important on the first read of the work. Let's say, don't worry if you don't understand anything, right? I've worked in this field for 8 years. I'll read a paper and I will not understand everything that they've done in my first read. I often read papers 3-4 times to properly understand everything. But what is important is, especially when you're new to an area or a technique or anything, make a note of the terms that you don't understand. It's very easy to say, okay, well, I'll just brush over that. Let's not look at that. But actually, a lot of those terms are really fundamental to you understanding the paper. And so do make a note of them, go and look them up, see what they mean, and that will really enhance your understanding of the paper. Okay, so once we've read it the first time, we've had a brief overview, we have a rough idea of what they're doing, we know their main question, we know where their question fits in what is known in the current literature, and we have a basic understanding of their approach. Okay, then on the second read, I always find it's easier to concentrate on the results. So that's the nitty-gritty of the paper, okay? And this contains all of the data that the authors base their conclusion on. Okay, so I like to think of this in terms of their conclusions should be effectively held up by the method that they're using, the data that they produce, and the statistical analysis that they perform on that. And what should happen is that these should all come together to give the conclusion that they present within their paper. Okay, and this 99% of the time, this is exactly what happens. But what you need to do within the results is you want to assess the quality of the data being presented. And what I find really useful is to write a summary sentence or a paragraph for each results section. And so this helps me. I mean, lots of people learn differently. I learn by writing things down. If I read something, it often doesn't go in my head that well. So I have to really write things down for them to go into my head. So what I like to do is I like to take each section, which is addressing a specific question, and then write out what question they're asking. I then write how they've addressed that question and the data that they've presented and what that is telling me. Okay, so the most important thing about this is, like I say here, is come to your own conclusions of what the data is showing. So a big thing is don't just read the paper, read the text, don't look at the figures and say, okay, well, they've said this, so it must be true because it's got published. Okay, there are errors in peer review. Papers do get published that probably shouldn't have got published. So it's really important that you read a paper critically. And again, try and say, okay, is their method appropriate? Do they present their data correctly? Have they performed the right sort of statistics? Have they even presented statistics? I'm going to go through this in a lot more detail in a minute. And then do you come to use those to come to your own conclusions on what the data is showing, and really do look at the figures. Okay, all of the data is in the figures, not necessarily the text. So that's the most important thing I would say is when you read the results, concentrate on the figures. Okay, and no, don't do that. We don't want to restart. So what I thought I'd do now, so I've mentioned about assessing the quality of the data being presented. Okay, so I thought what I'd do is go through some sort of key points in how you could probably critique the science paper, because if you haven't read a paper before, you might not know. I'm talking about assess the quality, okay, but you might not know how to do that because you haven't been given the skills. I think it's really, really important. And I actually think part of my degree as well, I didn't really get taught, and there's no real focus on how to assess the quality of a paper and the data. And I think, again, like I mentioned at the start, I think this is really great that Evie is putting these things on to enable you to sort of speak to people to try and get you to understand that. Okay, so the first thing to do is look at the journal that it's published in. Okay, so there's a load of journals out there. There's lots of journals that people just set up in their back garden that don't really have much rigor, and so lots of bad science does get out there. Okay, so what you want to know is it a reputable journal? So these will, a lot of them you may or may not have heard about. They'll often have their own their metrics, and so you'll be able to see by that if they're a decent journal. They're also often included in journal rankings and things like that. So if you have a quick Google of it, you'll see it'll be fairly clear the ones that aren't reputable journals. So that's the first thing to check. Again, has it been peer reviewed? So like I said, peer review is a really essential process for scientific publication. So many papers now, so there are these really, I mean, these servers are great. These are preprint servers. Okay, so what you can do when you submit your paper, like we did, you can put it in something called bioRxiv, and there's other preprint servers as well. And this puts it in the public domain, but it puts it in the public domain before it's peer reviewed. Okay, so as you can see here, so this is the title of our paper that we're currently in the process of publishing. You can see here, it says this article is a preprint and has not been certified by peer review. Okay, so it's really important to take that with a pinch of salt. Okay, so whatever they're presenting in their data, an expert has not looked at that and has not okayed it to go into a journal. Okay, so it's really important to look at this critically, even more critically than you would do a paper that has gone through peer review. Okay, so that's a really important thing to look at when you're looking at a paper. Okay, so going on to the, oh, has it done it again? Yeah. So what is the laptop doing? Sorry, guys. Let's go forward. The joy of technology. It's just frozen. Sorry, guys, I'm gonna come out again. And then hopefully come in again. So let's go down to where we were. Here. Hey, there we go. All right, sorry, I have no idea what's going on. Right, so like I mentioned, all sort of decent science, I always think a science should be hypothesis driven. Okay, so there should be a question that the researcher is trying to answer, and they should make that very clear to you within their paper. Quite often people will moan their paper's been peer reviewed and whoever's peer reviewed it hasn't understood, okay? And that's why they've told you that it's not very good. But actually, when at any setting, it's the same with anything, that it's your job to communicate it correctly and clearly. And if someone's misunderstood, then that comes back on the author, okay? So what you want to ask is the hypothesis very clear? Have they made it as clear as possible? And then look at their approach. So this is what I mentioned on that first read, is do you think that their approach is suitable for addressing that question? And then we go into the introduction. So how can we critique an introduction? Well, we can have a look and see and ask the question, do the authors outline what is known within the field? Okay, so you do see papers. I mean, I read one fairly recently that pretty much in the very specific field I worked in, that pretty much ignored any data that had been presented by anyone else because it didn't fit with what they said. Okay, and this got through peer review. It's out there, it's published, but they ignored pretty much most of the data that's out there in their introduction. Okay, so it's really important to see, unless it's something completely bonkers and new and there's no data, they should tell you what is known about the gene they work on or the process they work on or the drug they're working in or whatever. They should tell you what is out there, and there should be a number of references that you can then go and look at yourself if you're interested. They should show you where their question fits within the current knowledge. So this again is very similar to the first point here, is they should make their question clear, and they should show you or tell you the benefit of understanding their question and also how it would enhance the current knowledge in the field. And ultimately, is it well written? Okay, papers should be well written, it should be well communicated, it should be clear. If you start reading a paper and you think, God, this isn't very good, like it's normally means the data won't be that good as well. Okay, so that's another very, very important thing to look at, is the quality of the writing and the presentation of their data within the introduction. Okay, so we've got through the introduction. We're on our second read where we're going to concentrate on the results. And so three big things really is how have they presented their data? Is their data presented in a clear way to understand? What is their sample size? And have they used the correct controls? Okay, so these are three things that you should really be looking at and questioning when you look at their data. So for example, one question that you'll often get asked that you're taught back in GCSE is about sample size. And do they have enough biological replicates? Or for example, if you were to look at a clinical trial, do they have enough patients to address their question? And this comes down to statistics. And so what you should never see is an N of 2 or an N of 1. If you ever see that, then you can question why. Sometimes there are examples of very expensive experiments that are done that you can only afford to do a certain amount, but the authors should also address the fact and point out that they have a low sample size, okay? But this sample size will differ. Something that I do, for example, I might use 3, 4, 5, 6, 7, 8 replicates of a cell or a fly or something like that. Whereas obviously, if you're looking at a clinical trial, you'd expect there to be far more patients than that, for example. So this does vary, and you'll get a gist as the more papers you read in a field, the better understanding you'll get of what is expected for these sample sizes. Okay, so this does vary within the study. But most importantly is, are their controls suitable? Okay, so it's very easy to have something look different and you go, okay, that's great, that's done. But what you really need to show, science is about proving what it isn't as much as what is causing it. Okay, so you should be able to be fundamentally sure that what you're seeing is because of what you're doing, rather than it just being a technical error, for example, which is where controls come in. Okay, so quite often papers will nearly always have negative controls. A lot of the time they may have positive controls. Again, it depends on the work and the field. It's difficult to sort of sum this up that clearly, but negative control should always be there. And to go back to what I said about earlier, when we look at our drug response within our district 2 knockdown cells, we have this DMSO would be classed as a negative control. Okay, so what we'd expect is adding just DMSO to the cells has no effect, and that would be our hypothesis. Okay, and that's exactly what we see. But without this, if we didn't have this DMSO and we just added warp-mannin in a specific solvent, then we have no idea that it's the drug that's causing that effect, or just adding something to the cells in general. Okay, and this again is why we use, say, for example, you may not be familiar, but effectively the way we've knocked down district 2 is something called RNA interference. Okay, so we put RNA that specifically causes district 2 to be degraded into the cell. And what we have to do with this is our scrambled control. And so this is actually a sequence of RNA which doesn't target anything in the human genome, but it controls for adding something into the cells. Okay, so this is another example of a negative control. And so it's really important that they point that they have this in the data set, and that, again, the authors allude to the fact that they've used these negative controls. Okay, so these are two big things that you should look at when you're looking at the data and coming to your own conclusion. So this is a very, very sort of basic picture, but what you can have is people display their data in different ways. People have their own sort of preference on this way they present their data, but ultimately all graphs, for example, should have the same sort of three things. Okay, well, they should have the data, it should be clearly demonstrated. They should have the units on the axis. So for example, if you look on the right here, which is an example of a poor figure, for example, and a very rogue example of a good figure, it should have the units on the axis. It should have error bars, and these error bars tell you how variable their data set is. And so, for example, the larger the error bar, the more variation there is within the sample. Just because there's variation doesn't mean it's not real. Normally, the more, for example, if you look in a whole human system, there's normally a huge amount of variation compared to something like a fly, for example, which is genetically inbred, so they're all almost identical. And so there's far less variation. So just because you see variation doesn't mean the data is not good. Okay, but and then what the statistics look at is the variation within, for example, our mutant sample here, less than the variation between our mutant and our control. And that is ultimately what the statistics are telling you. So even though you might have a larger variation, if the difference between control and mutant is larger than the variation within the sample, then it's still a statistical significant difference. Okay, and so this is what, for example, often stats can be presented in a number of ways. It can be done using error bars. It can be done like this. It can just be done in tables. It can be done all sorts of ways, but there should be statistical analysis behind any conclusion that any scientist ever makes. Okay, so this should be very clear. So ultimately, we don't like figures like this, which don't have error bars, they don't have any statistical information, and they don't show units. Okay, so again, this is very, very basic, but there are papers out there that don't have this information within them. So be very wary of that if you ever come across it. Okay, so a very easy way to critique some data. But how do we understand? So I mentioned earlier that I said, make sure you look at the figures and don't take the author's word for it. And the authors will explain what each figure is they think is showing and how they've got to that situation. Okay, but all figures, all panels in all figures. So what I showed you here is just this figure here. Okay, so what you'll normally see is you read a paper and you get this really complicated thing looking with loads of panels, and you have no idea where to look because authors will very rarely show a single graph in a figure. So they'll be broken up into panels, and each one of those panels will be summarized in a figure legend, which I'll show you an example of in a minute. Okay, so all panels in all figures should tell you the sample size, so how many replicates they have. They should have what the error bars are, so whether might have heard of standard deviation, for example. They should tell you what that error is, statistical error. Again, they should have statistical information, so any images of cells or tissue should have scale bars. So for example, here you can see this black line, or this line here is telling you that this is 100 micromolar in this setting, for example, and that lets you make sure that the images have been scaled correctly, so any visual difference you can see is correct. And then this should all be summarized in that figure legend. And so I'll show you a figure legend now. Granted, I apologize for the amount of text within it, but what you'll see is this is the figure legend for this figure, okay? And each section will be separated. And what normally happens is, for example, if we look at C here, so it will give a summary of the data. So district light 2 wings in red are significantly larger than control wings in blue. And then don't worry about that. But what it then tells us is all this information that we're expecting to see. Okay, so we have our sample number, we have what our error bars are, we have our statistical information, and we have what the scale bar represents. Okay, so each panel will have its own little setting, and this will help you understand what is going on in the figure, and this should help you hopefully understand the figure enough to come to your own conclusions on the data and not rely on what the author has said within the text. Okay, and like I said, 99% of the time, what they've said in the text and what you come up with is exactly the same. It's just very rare occasions that you don't, but that's why it's important to really critique every paper that you read. So finally, when we get onto the discussion section, so we've read, we know where it fits in the field, we've seen their data, but what I like to think of the discussion as kind of putting your puzzle together. Okay, so throughout your paper, you've explained a journey where you've tried to address a question in a number of different ways. So you have all your puzzle pieces, and the discussion is where you bring those together, where you summarize how all of that data fits together, and then more importantly, where your data fits within what is currently known. So you add a piece to a bigger picture of a puzzle effectively. And it provides all the take-home messages. So the way to critique the discussion section of a paper, these are the questions that I ask myself anyway, is that are these sort of their take-home messages very clearly discussed? Do the authors address the limitations of their approach? So this is something that we're sort of drilled into us from very, very early on. Again, back to GCSE, you have to also, you might think your data is the best in the world, okay, the best data that's ever been produced, but the author should always know the limitations of their data, and they should say that in the discussion. And finally, do the results support their conclusions that they've drawn? And do they come to the same conclusions as I did? When I've looked at their data, do I come to the same conclusions? And so this is sort of how to ask yourself, it often helps to make sure you've understood the results properly, but also to make sure that they've presented their results in a way that is understandable and correct. So finally, I've kind of skipped over methodology because unless, as I've said here, unless you're familiar with the field, when you read this, it will seem very, very specific, okay? And a lot of it won't make sense. But ultimately, what any paper should have is they should provide information on every technique that they use. So there'll be a heading of, say, we measured fly wings, okay? So there'll be a heading of fly wings and how we measured them. But what we should do as authors is give you enough information to go away. If you had the lab and the capability to do so, they should be able to repeat your experiment exactly, okay? So then that is also a good way of making sure of understanding scientific rigor effectively. So you can say here, so for example, those cell numbers that I showed you on that growth curve. So this is exactly how we did it, the cells we used, how they were grown, what their food was, for example. Then we talk about how many cells we used, how much of that, so that RNA that I told you that we put in. So all of this, whilst this might not mean anything to you, if you're not experienced in the field, it gives you an idea that we are quite happy for people to take this away and try and repeat it. Okay, so this is what the methodology is there to do. So don't be daunted by it. It will seem very, very technical, but if you wanted to have a look, you could obviously have a look and just make sure that they have given you information on how to do everything. This is sometimes, depending on the journal, a lot of this can be in supplemental information. I haven't mentioned supplemental information. This is almost as important. A lot of the data that underlies the figures that are presented in the main text is in something called supplemental information. And you can always get this. So when you download the paper, this often isn't involved, but it will be available by a link on the journal website. And so quite often it's very, very important to also make sure that you have a look at the supplemental information if you really want to understand the paper properly. Okay, so I just thought I'd leave a summary here. So if we take our paper, these are the questions that I like to ask myself whenever I read a paper to come to the conclusions of whether I like the paper, whether I trust it, and whether I'm going to use that data to basically further my understanding and help me in my research and what questions I'm going to ask in the future. Okay, so this is stuff I've already been through. I think Evie is going to try to put together some sort of leaflet with everything. So yeah, so I'm not going to repeat it again, but this is something that you could, I say, the key things that I use to try and decide if I think a paper is a good paper. So finally, I'm just going to leave you with the current pitfalls that that you can come up with. So like I've mentioned, taking the author's word for it, okay? That is a common pitfall that people do use. Whilst obviously use the text to help you understand what they're doing. Try to come to your own conclusion as much as you can. Granted, your understanding of the field will limit this, but it is a good thing to try and do. A lot of people, myself included, I've done this in the past, is you read a paper once and you go, right, I've read that, I know that, that's fine, on to the next one. Okay, that's never the case. All right, so make sure you read a paper more than once to properly understand it. And looking up words you don't understand. Again, I've been very guilty of this in the past, where I've read a paper, I've gone, okay, I think I understand that. I don't really get those terms, but I think I've got the bigger picture, so we're all good. We'll move on to the next one. But do look them up. They're often really critical to understanding the paper. And also distraction. This is really easy. Granted, if you sit down and you try and read a paper, you might get bored. You might have something pop up on your phone, Instagram or Twitter or whatever. And suddenly, again, I'm 100% guilty of this. You look at it, you come back to the text and you sort of read a couple of sentences on and you're like, I don't know what's going on now. Okay, and then you have to go back. So really try, if you really need to understand something properly, make sure you focus on what you're doing. Same with anything in life. Okay, so to sum up, top tips for reading a paper: read the paper multiple times. The big thing is ensure you understand their main question. If you don't understand the question they're asking, you're not going to understand the rest of the publication. Again, I like to write myself a summary of each results section to summarize what they've, using points of the question they're asking, how they've addressed it, so the methodology and what they found and what I take that to mean. Okay, again, don't take the author's word for it all the time. Come to your own conclusions, which, as I say, 99% of the time will be exactly the same as the author's conclusions. As I say, critique the data presented. Don't assume because it's in the paper, it's great, okay? Because this will also help you understand the paper. The more you try and critique it, the more you understand. And ultimately, that's the critical thing here. Okay, so I hope that this has been some sort of interesting and useful, whether sort of hearing about some real life science is interesting, or sort of how I address trying to read a paper, is useful to you. Obviously, everyone will read a paper differently. They'll all have their own sort of ways in which they take the information in. So it really is a job of finding what's best for you. But say these are sort of some things that I do, and I hope that you find them useful. So I'll stop there, hand back to Evie. I'm happy to take any questions on what being a scientist is like, or what it takes to publish a paper again, or anything like that. So yeah, feel free to ask any questions you're interested in.
Lovely. Wow. Thank you so much. I was just thinking in my head, like, this would have been so helpful for me, this time last year, actually. But yeah, so basically now is going to be a Q&A session. So if you haven't already, you can use the Q&A box to put in your question. We've already got 7 in there already, so that's great. Again, you can anonymize if you don't want to ask it live, but don't anonymize yourself if you want to ask it live. You can ask, yeah, about anything, even if you just want some clarification on any of those slides or those points that Ben's made. So I'll start going through the questions now, but obviously keep adding as and when you want. And just to reiterate, yeah, those practical tips and hints, I'm going to make some kind of like pretty looking page with them all on and send them out, so you all have that, so you've got something to refer back to. And obviously this is recorded as well, so you could always come back to the recording and watch it if you want to go through Ben's explanations again. Right, okay, let's see what we've got. Okay. Oh, first question, how can medical students get involved in research? Yeah, go for it.
Okay, yeah, I mean, I think it's great. I think it's really important, and I've said this a number of times, where I think it's really important for us to understand sort of the clinician's way of thinking and how a clinician works, and also I think it's just important for clinicians to understand how the science works. And I think we really need to work very closely together, and the way to understand science the best is to get experience within the lab. So there's obviously, you can contact any PI. So I mean, I know here we have the IRP projects, which are in your 4th year. If you're a medical student, I don't know if you are. So that is when you get an opportunity to work in a lab and do a placement in a lab. And so projects will be advertised, and you can pick the projects that you find interesting, and then they'll be assigned based on a number of sort of criteria. So yeah, so I think the best thing to do is, if you're interested in a certain thing, then speak to your lecturers, or ask people in higher years that might have sort of been through the situation or have the opportunities. But yeah, if you have plenty of, if you're, again, I don't know if you're a medical school student here, but yeah, speak to your lecturers, and they're probably more than happy to try and help out as much as they can.
Lovely, thank you. Yeah, and I think just to add to that, I think I know from myself as well, I think it's going to be quite hard at the moment with COVID. Definitely. To get into the labs and do things like that, but there's research opportunities in terms of literature reviews as well. We're
currently very restricted, so we're only allowed 3 in the lab at any time at the moment due to COVID. So yeah, at the moment it's going to be very difficult, but hopefully when life gets back to some sort of semi-normality, we'll be able to have students back in the lab again, because it's great having them about.
Yeah, so that's great. So the next one, okay, hang on. Okay, this person also wants to be anonymous, that's fine. It seems ridiculous that after so much work, often journals ask researchers to pay a fee to publish a paper in their journal. Surely this is hindering dissemination of knowledge. How should the scientific community address this?
I entirely agree. Yeah, it seems ridiculous that you spend a fortune on your own work, and then you pay someone to present the work. So one of the things of those preprint servers that I told you about is sort of trying to get around that sort of thing. So I mean, a lot of the case, so for example, for my work, I'm currently funded by it's the BBSRC, so it's government funded, so it's effectively taxpayers' money. And so I am obliged to publish our work what's called open access. And so this means that our work is available to anyone who wants to read it without a journal subscription. So it basically bypasses the journal subscription, but the university, well, we have to pay it, but the university has a policy that they pay it for us if we're obliged to publish open access. So a lot of the time you will see that. Your university will hopefully have the rights to view a number of papers. So most university libraries will have subscriptions to all the big journals. You will obviously come across stuff that don't. If you've got any friends in other universities, often universities have different subscriptions. So if you have any friends anywhere else, you can always ask them. I remember doing that when I was an undergrad and needed access to a paper. I spoke to a friend and tried to get it through them. I don't know if that's the right one. I'm sure it is. But yeah, no, I agree fundamentally that it is wrong that we do all this work, and it's often paid by the taxpayer, and that's why the way I'm funded has that angle to it. And in the same way, I don't think charities have the same thing, but again, I think it's just as important for work funded by charity research, if not more important, to be honest, that work is able to be read by whoever wants to. I can't see how it's going to happen at the moment with the ways journals work, but I think I agree. It should definitely be something that we look at in the future to try and make it available to everyone.
Cool, lovely. And I think that applies, I think a lot of high schools and sixth form colleges and things, I think they have the same thing where they will buy the ability for students to be able to like look at different journals and things. Lovely, right. Okay, so I've got another question. So I am going to see. So this is a question by Abby, and I think she wants to ask it live. So let me just, Abby, I'm just gonna.
Technology is going to behave itself.
If it's going to behave itself. So, right, here we go. Where are you, Abby? There you are. Lovely. Oh, sorry, that's my doorbell. Right, Abby, yes, please ask your question.
Oh, yes.
Right.
I'm Abby. I'm currently in first year at BSMS.
Okay.
Do you think that in the future, gene therapy could be used to possibly create a treatment or even a cure for Perlman syndrome, or is that something that's not possible?
I mean, I don't know. I mean, I think gene therapy has a massive future. I think we need to do a lot of work to make it safe. So, for example, I told you about the CRISPR. There's been examples of the odd paper that comes out of places that are like, this was treated by CRISPR and the patient is absolutely fine. So, from a technical point of view, it would be feasible. You'd have to get it early enough, because the problem is you'd have to get it in the original cell effectively. Because obviously once the cell divides, there'll then be multiple copies. So I think this is where one of the problems is. There's also another problem is the off-target effects. So CRISPR is well known. There's a lot of work at the moment to make it more specific. So what we found when we did this experiment, this line was fine, but a number of lines have mutations in a lot of other genes. And so obviously that would be very dangerous in a human, but it could hit something else. And so this is why gene therapy needs to be really, really carefully considered and developed. And I think there's definitely a future for it. But yeah, I think a lot of work needs to be done to really make it accessible.
Thank you.
That's all right.
Good answer. What do I do now, Evie? Do I just stay?
It's don't worry, I'm gonna
go out.
Right, let me just okay, I think that's done it. I've got Peru in the background to help me, so I'm hoping he'll be able to. Right, I will go on to the next person. Let's see. Okay, this is another anonymous one. So let's sorry, there's just a little typo in it. How much should you rely on a paper that is published on a reputable website but is the only one on the topic? So cross-referencing with other papers' results would not be an option.
So yeah, so this is one of the examples I said where sometimes it's not possible. So again, I think if you were to use, if it's a reputable journal, if it's peer reviewed, for example, then obviously it's been through some sort of scientific rigor. So that's a good place to start, regardless of whether there's anything similar to it. And also, again, if you sort of have a look at the data they're presenting, and are they presenting it in a way that you think has enough scientific rigor, so sample size and etc. So even though you can't cross-reference it with other data, you might be able to view it on its own and decide if they've used the right controls, for example. I mean, it depends on the setting, but that's what I would try and address. As I say, there are examples of very unique papers out there where you can't relate it to anything else in the field or in any field. But yeah, that's the way I would try and address it anyway.
Okay, lovely, thank you. Lovely. On to the next question then. So this one, I think we might have to go back to the slide where you talk about the statistical significance, because this one is, what do the 4 asterisks mean above that graph?
So am I still sharing screen?
Not at the moment.
No? Okay, that's fine. So, I mean, I can tell you that the asterisks are often a way of showing level of significance. Okay, so is that, can you see that okay now?
Yeah.
Yeah. So if we go back to here, so this is an example. For example, here we've got 4 asterisks, and this is p is less than 0. So this, the way I do it anyway, and the way most people will present it, will be the degree of significance. So here you've got 4 stars as p is less than 0.0001, so very, very significant. You might have 1 star, so for example here, where p is less than 0.05, 2 star p is less than 0.01. So these are when you do the statistical output, you get a p-value. Okay, and the lower the p-value, the further it is from 1, the more significant it is. And so this is just a way of demonstrating the degree of significance on a figure.
Perfect. I hope that's made it clear to whoever asked that. If anyone else needs anything clarifying, please do ask.
Let us know.
The next one, another anonymous one, I might actually be able to help with. Would the advice you have given be useful for creating posters? I've never created a poster before, so I'm quite nervous about it. And I would like to say we're hoping to watch this space, basically, because another colleague of Ben's, Dr. Ollie Rogoyski, I'm hoping to have him in January to talk specifically about this topic of how to create a scientific poster and how that process works. So watch this space.
Absolutely. So it's very, very important. And as I say, I don't know what stage, if you're doing medicine, I don't know what stage you are, but you do have an assessment on that, where you're given a paper and you have to present a scientific and put it in a poster format. Again, not to sort of jump on Ollie's parade, I'll just say that the main thing with the scientific poster is do not put too much text on there. Okay, I find myself writing the same feedback every year, which is there's too much text. Okay, so use figures and use pictures and try and make the poster you need it to be visual. Okay, it's to grab someone's attention. So what we do when we go to scientific conferences, some people present talks, and then others, normally the more junior researchers, so myself included, will present posters. Okay, and this is a way of us standing there presenting our data to the experts, and we work through the poster. And so from there, we want to grab people's attention using it. So just seeing blocks of text is never going to grab anyone's attention. So that's the main thing I would say. But I say, hopefully there'll be someone in the series to actually go through that in much more detail. Because I think it's a really, really important exercise. Again, like I mentioned earlier, communication of science is so important. And a poster is a very good way of doing that at scientific conferences.
Yeah, maybe it might help if anyone else wants to add a question, if you could just quickly say like what year you are and like what, like if you're a student, if you're at college, or what it is you're doing, just it might help with a bit of context as well. So this next person is Human, and they've got a question and they haven't anonymized themselves. So I'm assuming, are you happy for me to put you live? Let's see. Are you there?
Yes, hello.
Hi.
I'm Human. I'm a first-year medical student at BSMS, and I just wanted to ask, so you said that like one of the indications of a paper being like credible or good to read is like the journal, but before that is the fact that a certain paper is in PubMed or like these sort of websites, is that a good indication or not?
So a lot of, as far as I'm aware, I might be wrong, a lot of, if it has a PubMed ID, it's normally a reputable journal. But there might be examples of not, and as I say, don't take my word for that, but normally PubMed is a very good place to start. PubMed or Web of Science are the best two really search engines, to also databases, as it were, to use to look for papers. But yeah, so in general, a PubMed ID does help. Obviously, like I mentioned, there's lots of different types of journals, right? And the journals vary in how they're ranked. Okay, but anything on there is normally a decent journal, and it depends on your field as well. So I mentioned this is one of the reasons why I said impact factor is a bit all over the place, because it depends on your field. So the best paper, the best journal you might, unless you have the Science and Nature or the Lancet, for example, when you go below that, if it's quite a specific field, then actually the impact factor might be lower, but it doesn't mean it's not a good paper and it's not been done properly. yeah, I hope that answered your question to some extent.
Thank you. Yeah.
Yeah, cool.
Brilliant. Thank you so much for that. On to the next question. What have we got? James, where are you, James? I'll try and let me find you. This is like this is really stretching my tech knowledge. So I really apologize. James, there you go. It's just coming up. That's it. If you can
Hi, sorry, can you
oh,
hear
thank
me?
you.
Yeah.
Yeah,
Perfect.
unfortunately, I didn't realize the rule was anonymized if you can't put your camera on. I've got, I was just doing my laundry, so I have laundry drying in my room, so I will spare you that. Sorry, my question was just, is there a process to report any papers that you deem unsatisfactory with regard to sort of their trustworthiness or legitimacy? I know you said 99% usually are fine, but with that 1%, is there a way to sort of report that?
You'll see. You would have heard very famous examples of papers being retracted, and that normally comes from the scientific community. So if people have tried to repeat something, sometimes papers can be retracted due to sort of very legitimate things. So it could be a genuine mistake that's been made, and people retract them. Other times, there's very famous examples such as certain vaccines, where people have gone to prison for it. But this is normally called out by the scientific community. So what normally happens, as far as I'm aware, is people will communicate with the journal itself and say that they have a problem with the paper, and I think the journal sort of takes it from there, as far as I'm aware. Again, I might be wrong, and there might be other reasons, but in general, I'd imagine if you've got a problem with something, to contact the journal.
Oh, brilliant. Thank you very much.
That's okay.
No, I'll leave you to get on with your laundry, James. Okay, last couple of questions now, but you can still send in some more if you want to send in some more. So this one's anonymous. So for untrained eyes, how would you recommend working out what figures results are significant and what's not without taking the author's word for it?
Okay, so as I say, so in terms of an untrained eye, again, I think it goes back to, like I mentioned, looking at if they have those key things within the figure, in order to understand the figure. In terms of, sorry, can you repeat the second bit again, Evie? I've completely forgotten where I am. Getting old.
Hang on, where is it? For untrained eyes, how would you recommend working out what figures results are significant and what's not without taking the author's word for it?
Yeah, okay. So I mean, as I say, what it should be is that the figure makes sense to you, and having read the reason they're interested in it, does the result. So for example, again, a very basic example, but if I say here, our hypothesis is that dystrophy like 2 ingrows growth, and then here we see these bars are larger than these bars, and we've given some statistical information, and we've given the sample size and everything like that, then I think that would be a reasonable assumption to say that, okay, this has been done with correct sample size, scientific rigor, etc., and it's addressed the question that they're asking. I think that's the biggest thing. As I say, I'm fully aware that the less experience you have, the harder that is to do. And then say the more you read papers, the more experience you get. I mean, it's the same with writing. I was quite frankly awful at writing when I started my PhD. But through reading more and more papers, you get better and better at it. And you get better at understanding the figures as well, just by sitting there. So just persevere a little bit, and hopefully things will start to click. But yeah, I understand without necessarily a trained head, it is far more difficult. But yeah, I think they're the key points that I would use anyway. So I hope that answered it to some extent.
Lovely, thank you. So the next one looks like this person is a graduate studying medicine. Do you think it's possible for grads to get involved in research in first/second year of medical school? Okay,
so it all depends on the lab. I mean, obviously you're very, very busy in your first and second year. So I think a lot of people might suggest to maybe at least during term time to not spend too much time doing stuff other than your studies, as you are very, very full on, as I'm sure you found out. But in general, there's often summer projects which are advertised. So for example, if I go back to Evie, for example, I mean, she's granted a BSc undergraduate doing biomed.
Yeah.
But we advertised for a summer student because we had a project, and so that often goes around. And I think the medical school also does the same thing. But as I say, if you have any lecturers, for example, that you either know or you see what work they do, if you're interested in it, then speak to them. I mean, obviously it depends on funding, because science is expensive. So whilst people would like to have volunteers coming along and helping, it also depends on the cost and what the lab has at the time. So you would never guarantee it, but there's no harm in asking them, because they might know of someone who's looking for someone. But yeah, I'd probably try not to do it during term time, because as I say, you are very, very busy with your studies. But the summer might be a good opportunity to try, and even if it's a couple of weeks, just seeing how a lab works.
Yeah, and I have to say, like, I know that there are students in my year that they just emailed a bunch of the lecturers or clinicians, people that we kind of come into contact with their kind of idea for a project, or just kind of where should I go if I want to get involved in it. So it never hurts to ask. All that's going to happen
Absolutely.
is they either don't reply for months, or.
And we're very, very lucky. I'm not just saying this. I mean, most of your lecturers, at least the scientific based ones, are all in the building that I work in, and they're all really, really lovely people. Like, don't be scared by them. Like, I'm sure they're happy. I'm sure next time they're going to come down, I've had 300 medical students on my door and tell me off. But yeah, drop them an email. I'm sure they'd love you to show an interest in their work, because ultimately we just want to get praised.
I love it. Yeah. Great. So this next one, also, so last couple are anonymous. If you would have to research a field X, how would you go about it? Where would you start? Okay,
so what, in terms of just starting somewhere completely new, or?
I guess so, yeah.
Yeah, I mean, it's difficult, right? So the way I got onto Digital Light 2, for example. I mean, I started working in Sarah's lab on something completely different, and then I was waiting for an experiment to finish, and then I started playing around with this, and then it ended up being sort of the stuff I was doing got thrown in the bin, and then I've concentrated on this for the last 7 years. So a lot of it happens. I mean, it sounds like a cliche, but a lot of science does happen by accident. But obviously, to do some work, you need to have some money to do so normally. Again, it depends on the setting. So molecular biology, like what I do, is very, very expensive. And so we need grants and we need funding to do it, and we need people to think what we're doing is a good idea. But obviously the fun thing is, whilst it might not be completely what you asked, the fun thing is the creative thinking. Okay, and we say, okay, well, we're sort of working on this, but also what else could we play around with in the lab, and what else could we do? And so that's part of it anyway. Is just being creative and thinking what other experiments could we do, and that comes from reading as well. So reading papers, we get all my ideas largely for work come from reading papers and think, okay, well that sounds cool. So how can I apply that to what I do? But yeah, it's quite difficult in my field anyway to sort of start from complete scratch because of the cost required. But yeah, I think the fun thing is sort of doing multiple things. I hope that helps to some extent.
Yeah, and I think your earlier point, when you said looking at the reviews, article reviews on different papers, that really helped me when I started my summer project. I think you just shoved 5 different papers, including
Probably
a couple of reviews on my desk, and there you go, that's your reading for the week. So yeah, and the reviews, especially systematic reviews, I found really, really helpful, just to if you're going into that field, you get to know the scientific gossip of what's going on.
Definitely. Also a good place to find original papers as well. So the review will have all of the original papers from which it's taken its conclusion, from which it's written the review. So it's a good place to find other papers as well to read in the same field. It's often how I do it.
Yeah, definitely, especially when it comes to writing an essay or something.
Yeah, absolutely.
Find my whole reference list.
Your whole reference list is in a single paper. Yeah. I've also done
that. Oh, good. I'm glad it's not just me.
There you go. Wow.
What have I got here? Thank you for this talk. Where would these summer projects get advertised? I can't even remember where mine was advertised.
I mean, yours we sent round to the specific courses that we thought would be interested in it, is normally what happens. So either it depends on how it comes about. So sometimes we'll always say over the summer that we would like a student to work in the lab. And so we send that round to the respective courses that we think is relevant to the work we want to do. And then we'll get applicants, then we'll sort of interview them and decide on who we're going to take. And then normally we have a project in mind for them. Again, that will depend on the lab enormously. So some labs, they might appreciate you coming up saying, sort of, I know you work on this. I think I've sort of read about this. Is there something we can do about it, or we can do together? But in general, I think they're emailed round. So I mean, they might. I don't know if they pop up on the website or anything, but in general, the way we do it anyway, and that's all I can really comment on, is we email round to course heads, course leaders, and then they send it to their mailing list. But yeah, I don't know how other people do it.
Me neither. And I suppose, you know, you could always be proactive about it and just email the heads of the labs yourselves.
Yeah, it comes back to that, definitely.
Yeah. Okay. So I think we have one more question and it's going to be live. So Peru, would you like to ask your question?
Hi, thank you very much. Thank you very much, Dr. Towler, for
That's all right.
Talk. And I also want to pay tribute to Evie for, as you say, organising a fantastic series, which would have been so useful when I was in first year as well. My question relates to abstracts. Sort of, if especially if you're a young researcher and you've got a vast amount of data, how would you advise in terms of picking out the most important bits to include in your abstract? I'm sure it depends on what field you're going to be talking
absolutely.
about.
You'll also see different fields write their abstracts differently. So sometimes you'll see, I think a lot more medical journals and everything will have sort of their sort of introduction, their results, their methods, their discussion, all outlined in separate sections, whereas that's a lot rarer in sort of more basic science, where it's all summarized in a paragraph. So, but in general, you just want to give reader enough to get an idea of if the paper's of interest to them. So if it's in the right field, so a couple of lines of introduction effectively. Telling them what you're working on and where it fits. And then normally we just pick the big take-home messages. So what are most people going to find interesting? So what results are the most amount of researchers going to find interesting here? And sort of give them sort of a taster for what might be in the paper. But yeah, I think, and if you do the work, it becomes quite clear to you what those results are. It's a bit more difficult if you're given a paper and told, can you write an abstract for this? But it does become clear to you when you've actually done the work yourself.
And just a follow-up question, because I know quite a few med students actually go to conferences to present their research. Do you think there's a different art to submitting a conference abstract to what you may do for a paper?
In terms of the field that I'm familiar with, not really, because again, you're just giving a summary of the work. I mean, you might change it. So some people write the abstract, if it's for a talk, for example, will more like, I will present this data, whereas you obviously wouldn't do that in a paper. That's the only difference really. In general, it's still the take-home messages, because quite often you have to, if you're interested in talking in a conference, I mean, it depends again for the conference, but there'll be a set of invited speakers and then there'll be slots open for people who, if they want to. So you put an abstract forward and then they decide whether they want you to talk. And so you might do it, you have to sell yourself in either that or the paper anyway. So yeah, in our field, it's very, very similar. But again, I don't know about others. It might change depending on the kind of work you do.
Thank you very much.
That's okay.
Okay. If Ben, are you still happy to carry on for a few more minutes? Because I've just had a couple more questions come through. The first one, any resources you know of recommend on writing in this format? Would you mind just, the person who wrote that, would you mind just clarifying what you mean by that?
So sort of how to write scientifically sort of thing.
In this format. Yeah, I'll wait to see if they ping up with something. Let's go on to the second one. For statistical analysis, should we learn how to use the softwares, or how can we generally do that?
Okay, so if you're in a lab, or a research group in general, obviously a lot of research isn't lab based, then they will, whoever you're working with, unless you're in a group already, but whoever you're working with should understand the software and should teach you how to do it. I know, for example, with Evie, we showed her how to use the software we use for statistical analysis. I mean, the stuff we do is very simple. Some statistics is very complicated. Like, I'm very good friends with the statistician at BSMS, and he does far more complicated stuff for far more complicated studies. But we often have if it is something like that, then we'll have a specific statistician who's able to advise us on the work. Otherwise, the group that you're in generating the data should teach you how to use that software. And yeah, there's specific softwares to do so.
Yeah. Okay, lovely. Yeah, and again, I'm really hoping to run a workshop with the friend that Ben talked about on statistics. So if that's something you want to learn more about, and I'm thinking this applies for anyone, of course, but for your medical students with your IRPs, your research projects, if there's some particular statistics you would really like a little workshop on, please could you let me know, because then I can try and arrange that basically. So thank you. The other person hasn't got back to me. So the question is, any resources you know of recommend on writing in this format?
So, I mean, I would interpret that as is there anywhere where you can learn how to write scientifically, I suppose. So again, there are courses that the university run. I'm not familiar with which ones are open to medical students or the undergraduate students or postgraduate students and all this. Again, I would probably, if you have an academic advisor or a tutor or something and you're interested in that, then maybe I would go through them if you're a student here. And they should be able to point you in the right direction, because there is an art to writing scientifically. And as I said, I was awful at it when I started. I mean, writing has never been my strong point at all. But I've learned over time, and I learn this, again, probably Evie will tell you about this. So we always, whenever we have students to supervise, we always sit them down and we go through roughly how to structure each paragraph. And each paragraph, I always think in scientific writing, each paragraph should stand alone, or each section should stand alone. And again, going back to what I said about writing a summary of each section, that's how I like to try and write each section. So each section, I want them to know the question I'm asking, how I'm addressing it, what I've found, and briefly where I think it fits. And I think if you try and summarize each section like that, then you should be along some decent lines to writing something that flows relatively well and is understandable. But yeah, speak to whoever, an academic advisor or something that might be able to point you in the right direction if you want to get involved in anything like that.
I remember writing my dissertation and it would come back draft after draft after draft, and it would come back like just so many comments, like even like little things like, oh no, make sure you use a capital here and a lowercase here to represent different things and stuff. So it's like, it's a lot.
Again, it's a learning process. It's 100% a learning process. And yeah, you get better as you do it.
Yeah.
I still read things. So for example, I write a paper and I write the paper and give it to Professor Newbury, my boss, and then she writes it and sells it and makes it far better. But yeah, it's a learning process.
Lovely. I like this one. Okay, I'll make this one the last question. How did you feel when you finally completed your PhD? Does it feel weird being called Dr. Towler?
Yes. I'm still not used to it. And it's still weird when you see your post. I mean, to be fair, I've only changed about half of the stuff. I haven't changed all of it over. And that's been, what, 4 years? But yeah, no, it is still weird being called Dr. Towler. When you finish a PhD, it's a real sense of achievement, because it's hard work. And anyone who's ever interested in doing it, I would always say it's the best thing I've done. I absolutely loved it. But you really do have to be committed to it, and you have to want to do it. It's very much like a medical degree, right? It's not a walk in the park. You have to really want to do it, because that's what keeps you going. I mean, the data I presented here is all the good stuff, right? 9 out of 10 experiments don't work. And you never see that data, you never see that work. Or, for example, those pretty pictures I showed you of the colourful discs, that took about 6 months of work to get it working. And it looks really beautiful when it's working, but it is hard work. So yeah, so it was a real sense of relief as well, and achievement. And yeah, it definitely still feels weird being called Dr. Towler.
Wow. Okay. If anyone has any more questions afterwards, you can email me. I'll make sure my email's put out on the thank you email afterwards, so you can email any questions and I'll forward them on to Ben, or get them all back to you. I think you'll also be emailed out a survey link to fill out, just to see how it was, like good things, things we can improve on, anything else you want to see in future. That's what I'm really keen on.
Please let me know if there's anything. Yeah, sorry to interrupt you. Please let me know if there's anything that you'd like to have seen that you didn't see, or anything that you I think you won't think I went through too quickly or anything like that, because again, like I said with writing, and the same thing with this, is a learning process. So do feel free to let me know.
Brilliant. Okay. Thank you so much. I would also just really quickly like to thank Peru, who is the OHP president of the society, and Laura as well. They were massively instrumental in helping me because I do have flaws myself, and they thankfully make up for it. So thank you so much. And obviously, thank you so much to you, Ben, for giving your time tonight to talk to us bunch of students. I hope, and also thank you to everyone that's stayed right to the bitter end. Well done. Oh my gosh. Oh, there's some thank yous coming up in the chat as well. That's everyone. Really sweet. So yeah, big thank you to everyone, especially to you, Ben. And yeah, watch this space. I want to run more. If there's anything you want to see, anyone you want to see, like